Good evening, ladies and gentlemen. We are about to start now with our roundtable discussion, which is kind of a special event in the frame of the Senckenberg Messel Conference for this year. And we are actually very pleased that a bunch of leading specialists in the field of primatology and paleontology agreed to participate in this special event for this evening. And actually, I would also thank all of you and Erik Delson, who is going to be the moderator for tonight. And I think we should directly start. So I will hand over to Erik, and he's going to start with an introduction. Thank you very much.
Good evening. We're going to work today on a rather structured schedule so that we fit the most information for you and us into the time available. So the first step is for each of us around to introduce ourselves so that you know we can't see you because of these lights in our eyes, but you can see us and have an idea that you should be able to read the labels. We can introduce ourselves to you and explain what will happen. So in brief, there will be a series of topics growing out of the discussions earlier today and previously. And for each of these, I will pose the question and try to introduce it for the audience, and then ask one or two of the speakers to give us 3 minutes or 2 minutes discussion of that topic, followed by choosing several of the other members of the panel to respond, perhaps to support or to argue against the discussion that has followed, taken place. Then the original speaker will get a minute or slightly more to respond briefly, explaining why his view was right after all, and then we'll start the next topic. And if we have some time at the end, we can have some more general discussion. We'll see how that goes. So basically, my name is Erik Delson. I'm at City University of New York and the American Museum of Natural History. I have not worked on the Eocene since 1969-70, but I am rather a generalist, studying something about almost all primates, from plesiadapiforms through to the origin of modern humans, with a specialty on Old World monkeys, which probably will not be mentioned again today. And thus, I have an idea about the topics of discussion here, but don't have any specific ideas of my own to push. Let me start now, and I'll turn to my right and let the first speaker introduce himself, and we'll just pass around the table this way.
Thank you. Is this working for everyone?
Seems to be.
I'm Alfie Rosenberger. I'm at Brooklyn College in New York, actually part of the same university system as Erik, also at the American Museum of Natural History. Also like Erik, the topic probably closest to my heart is platyrrhines, not cercopithecoids, but a topic that we didn't discuss at all today, the origins of platyrhines, and we probably won't talk about it much tonight either. Of course, I'm interested in anthropoid origins as well. Sufficient?
Sure. Saves a lot of time. Unless you want to make an argument for some point that you'd like to make later, which was the idea.
Oh, I can do that. Sure,
Why
I can
don't
argue.
you do that?
I'm sorry that I don't follow the rules all the time. A lot of the discussion has really centered or indirectly focused on anthropoid origins, clearly. That's one of the things that we've been talking about. We were asked to talk about this in some ways. Actually, the origins of higher taxa, but anthropoids became a focus because the Darwinian controversy, so to speak, sort of focused on the issue of anthropoid origins. I think that we have a long way to go still before we can really give a high-resolution answer to the question of where anthropoids came from. It's a difficult question. we want to know, but I think we're lacking in many very important things.
What's an anthropoid?
Pardon?
What's an anthropoid?
Oh, I don't know what an anthropoid is, and that becomes the first question.
Today, what is an anthropoid?
Yeah, the definition of what anthropoids are. Yeah. Maybe we
explain that to the audience. That's what you're here for.
I'm sorry.
He was a student of mine 400 years ago, and he still needs a little help.
Yes. But we do play together very well. Anthropoids include the New World monkeys, the Old World monkeys, apes, and apes include, well, hominins, us, our relatives, and our ancestors. So this is a major group of primates, a very successful group of primates, with lots of interesting characteristics, the adaptations that we all share in common that make us special. Many of those were inherited from the last common ancestor of all the anthropoidea. And it's just a fascinating issue. It has been for a very, very long time. But as I was trying to say, I think it's a very difficult question. And it's difficult not only because of a dearth of fossils, the right kinds of fossils, but also because we do have problems methodologically understanding or grappling with this type of a question. And as you've seen today, there are a number of different approaches to analyzing data. And because of differences in our philosophies that really feed into differences in our approaches to things, it's inevitable that there will be differences of opinion. That doesn't mean there isn't common ground, but it makes things very, very complicated. And I think we'll be in that situation for a long time to come. Is that okay?
Sounds great.
Thank you.
Thank you very much. Okay, Wighart.
My name is Wighart from Koenigswald. I'm emeritus from the University of Bonn, and my relation to Messel is just a period of 10 years in my career when I was at the museum in Darmstadt, and we were excavating in Messel like the Senckenberg Museum did. At this point, I wanted to mention one thing, which I think is really important for this conference. It was Jens Franzen who drew attention to that site to all of us. We didn't know about it, in fact, and he forced us, just long time before I got that position in Darmstadt, to look at Messel. If he wouldn't have done that, we might have lost that site, for there were not too many others who really imagined the significance of that site. So, this is credit to him. On the other hand, if people are working together, they do not always agree. And I'm not a paleoanthropologist, I'm not a primatologist, so we have some discussions there, but that's not very important. I do not agree at all. The theory he demonstrated here, that we have volcanic exhalations in Messel, for the volcanic phase was, according to the other investigations, 700,000 years or even more in the past. So there's a huge gap of time. And so I think that Darwinius, we want to talk about, died of volcanic exhalation when he was sitting on the ground. I think that is not the proper picture. There are many other possibilities to discuss, and I think that has to be kept in mind, that it is not only one single solution for all the various things. We saw that poster because of the dead turtles, and this was a very elegant explanation for one aspect. But by the way, I worked a little bit on the Messel mammals and described several of them, and so I may contribute a little bit even to the primates of Messel. Thank you very much.
Thank you.
Okay, my name is Xijun. I'm from Beijing, and I'm a research professor of the Institute of Vertebrate Paleontology and Paleoanthropology. And I'm also a research associate of Carnegie Museum of Natural History and American Museum of Natural History. Now I basically study the origin of a primate. I pay more attention with the earliest primates and their relationships to other primate-like creatures. And I also interested in the early diversification of primates. And I think Erik asked me to say that we use your primate today. And actually, I think primate is primate, your primate, eco-primate, and all the others like flying lemurs, tree shrews, and some extinct Paleoanthropology, they are primate relatives. They are not primate. And the earliest primate, I would say it's some creature like Tehadina attica. And that's, fortunately, I published that fossil a couple of years ago. It's a tiny, tiny primate. The skull is about 2 centimeters, and it has a rounded brain case and have quite a lot of teeth. Looks like insectivora, but have convergent orbit and with a post-orbital bar. That's a typical primate feature. And for the early diversification of a primate, I think probably the so-called striped sireni and the hoplorini diverged very early. Yeah, that's it.
Thank you.
My name is Erik Seiffert, and I'm an associate professor at Stony Brook University in New York. Most of my paleontological research has been working with Elwyn Simons in the Fayum Depression in Egypt, which for a very long time has been arguably the most important area for recovering remains of early anthropoids, but fairly derived anthropoids close to the crown group. So I've been doing that for the last 11 years or so, but I'm also more broadly interested in patterns of mammalian evolution, and particularly what has happened over the course of the last 100 million years or so in Africa, because it's a major kind of a blank in our knowledge of early mammalian evolution and definitely primate evolution. So I've been involved in the study of anthropoid origins, but also kind of an unexpected byproduct of our work in the Fayum is that with the discovery of the oldest land mammal site in Fayum, we also discovered the earliest, I consider, definitive crowned strepsirrhines. So the oldest relatives of the living lemurs and lorises. And so that has, I think, helped to kind of fill out the picture of strepsirrhine origins, which was previously that the oldest fossils were only about 20 million years old. Now we have fossils that are about 37 million years old. And with other work that's been going on in North Africa with people like Rodolphe Tabous and Laurent Marivaux and others, we're starting to find a lot of fossils that we think are probably stem strepsirrhines. And I think that isn't something that we've really talked much about during this conference, but that's actually a record that's becoming quite rich and that I think has important implications for understanding the origin of strepsirrhines and how adaphiforms fit into that whole picture. And so hopefully that will also be a topic of discussion. Another point I'll make, and I think we're all going to talk about it, is that I think that a lot of the reasons that we disagree about the phylogenetic relationships among the crown primates is because we might not have a single stem primate known in the fossil record. And so based on the results that Xijun has presented, perhaps all of the potential stem primates, the plesiodapiforms, could potentially all be dermopterans. And so that could legitimately mean that we have no stem primates. And with no stem primates, we can never figure out what is primitive and what is derived. And that's
Could you describe for those members of the audience who don't know the difference between a stem and crown taxon?
So when we're talking about the crown group, we're talking about all the living taxa and their last, all the descendants from their last common ancestor living or extinct. And then you have the stem lineage, which is entirely an extinct group, what we'd call a paraphyletic group at the very base, outside of the crown group. Finish there.
Okay, thank you. Gregg?
Hello, my name is Gregg Gunnell. I'm the director of the Division of Fossil Primates at the Duke Lemur Center in North Carolina. In fact, I sort of connected with what Erik talked about just a minute ago because I've taken over the position that Elwyn Simons held there for nearly 40 years, and it is a daunting task, I will say. I, after a failed attempt at becoming a paleoanthropologist, I moved on into looking at earlier primates and cut my teeth in the Badlands of Wyoming, the western part of North America, looking at actually collecting and trying to learn something about early primate evolution in North America, which kind of has led to becoming interested in many aspects of primate evolution. I will say, before I go on any further about primates, that I have attempted very hard to get away from primates. As you saw the other day, I talked about bats, and they've kind of become my first love these days, but I'm being dragged back into primates. And so if I ever had a dog in this fight, it's probably been long dead. So I will sort of try to go along and see what I can add to this discussion. The things that interest me now, I think, are less about specific sort of relationships between particular groups of animals or even particular groups of primates from various places, but more trying to understand the ways in which we can define things like origins. We throw the word origins around a lot here. We talk about anthropoid origins, we talk about primate origins, and I think there's many ways that one can assess origins, and I think this is something that's become of a bit of interest to me. Also, I think having moved to the Lemur Center and having had the chance to spend some time with lorises and lemurs, these strepsirrhines that everybody likes to call them, I've had a sense that they're very fascinating to watch. Their behaviors are very fascinating, but they really appear very primitive compared to the things that you might see in a zoo of a monkey or a great ape or something like that. And so I really don't know where they fit. I would be comfortable believing that they split off from the main lineage very early in time, and I think this is something we might want to get back to talking about at some point in this session. I'm going to stop right there and hand it off to Chris.
Thank you.
Thanks, Gregg. I'm Chris Beard. I'm a paleontologist at the Carnegie Museum of Natural History in Pittsburgh, Pennsylvania. I consider myself a mammalian paleontologist and a field paleontologist, not only by training but by desire. I think that paleontology, especially here in the early 21st century, is a discipline that is driven by new discoveries in the field. The episode about Darwinius is one example of how a new discovery has driven a great deal of research and discussion. But there are many others that fit in the same line, and that's why we all go back to the field year after year searching for that elusive fossil that's going to tell us the answer that we're all trying to find out. In particular, I'm interested in a couple of things. I'm interested, like many people up here on this panel, I'm interested in origins of major taxa, especially major taxa of primates. The order as a whole is a big challenge because, as various people have pointed out, we don't have obvious stem taxa that can polarize primate characters and give us a good idea about evolution within the group. Anthropoid origins, being a little bit closer to that terminal taxon Homo sapiens, is even more controversial and equally challenging. I think a good analogy is to think back to 100 years ago, to the early 20th century, when we knew virtually nothing about Australopithecines and fossil hominins, and great paleoanthropologists of the time like Sir Arthur Keith were gesticulating and pontificating about human evolution with virtually no data, and they came up with all kinds of interesting schemes, almost all of which turned out to be wrong. Anthropoid origins, I would say that we're not quite at that stage, but compared to the field of human origins today, anthropoid origins is vibrant scientific ground, because basic questions like where did anthropoids evolve, when did anthropoids originate, and from what, and how did that macroevolutionary transformation from a relatively primitive primate of some ilk to something that we could all eventually say, okay, that looks like a monkey. These are very much open questions. So, a big question for me is why isn't, every other week, why isn't NOVA doing a special on anthropoid origins instead of some hominid thing? But anyway, those are some of my ideas. I guess the other thing that I would say, though, is that we have, even though they're great lacunae, we know some things. Giant advances have been made in the field of genomics. I think we can all agree that we know now what the relationships, at least among higher level living primate taxa, are. And those relationships strongly constrain what we can do with our fossils. It doesn't completely constrain us, but it does strongly constrain us. And so those are interesting things to ponder. I think I'll stop with that.
Yes, please. Thank you.
So my name is talking, yes. My name is Marc Godineau. I am a professor at the École Pratique des Hautes Études in Paris. I spent 30 years, more than 30 years of my life studying primate fossils, 17 years at the university in Montpellier, and then I continued in Paris, and I specialized on Eocene primates from Europe. Now, for the audience who doesn't know exactly where the Eocene is, it's a period starting at 55 million years ago and stopping around 34 million years ago. So it's almost 20 million years of time. And there are quite a number of primates pertaining to different groups. And so it gives the opportunity to study a good deal of evolution from small, primitive forms, and to see the development, the progressive specialization of locomotion, dentition. And it gave me a kind of experience of history within some groups. And I like this historical approach to the fossil record, to evolution, to the evolutionary process. Along the way, because one great fossil was found, a complete hand was found in the Quercy phosphorite, I spent between 5 and 10 years working only on primate hands, comparing this fossil to living primates, making detailed study of the carpus and all that. And along the years also, I became interested in broader questions of primate evolution. I had liked 15 years ago to interact with colleagues who were starting to do some molecular phylogeny. However, this very nice history of European primates that we have stops suddenly at 34 million years ago, and in fact, all these primates for which we have a very interesting record cannot be the ancestors of the living one. They became extinct because of big climate change, and so the ancestors of the living primates are coming from elsewhere, and in particular from Asia or from Africa, from region from which we have a much less complete record. So one reason why there is still a lot of debates concerning primate phylogeny and primate history is that where we have a very rich and good record, those primates became extinct a long time ago, and the geographic areas on which the living primates evolved have a much less rich, have a very poor record in part, and that explains part of our debates. And I have some ideas that I will be happy to share on different aspects of these debates. Thank you.
Merci.
My name is Philip Gingerich. I'm a professor at the University of Michigan in the United States. As a student, I was very fortunate to work with a professor who did field work every summer, and especially in the Paleocene, collecting Paleocene, what we called then Paleocene primates. We collected them by the dozens, and I remember asking him after a long day in the field how these samples we were collecting were related to each other. And I remember being terribly disappointed by the answer because it was clear he didn't know. And I thought, if evolution is true, we should be able to show people sequences of change through time, and that then became my PhD thesis to study a group called Paleocene primates called Plesiadapus and its relatives. As an aside, many of us don't think those are really primates anymore, but it was a good chance for me to become familiar with the record before the beginning of the Eocene that Mark talked about and that we're focused on here today. When I finished my PhD, I came to Europe on a NATO postdoctoral fellowship and was very fortunate to work at the University of Montpellier, where there are good collections of fossil primates, and I could easily visit museums all through Europe to study the group I was particularly interested in, which was the adipoids, the larger of the Eocene primates, and it gave me a chance to become familiar with the fossil record of what was there. I was struck as I studied them repeatedly, I was struck at how similar they were to things that my former PhD professor, Elwin Simons, was collecting in Egypt. And so I've been focused on the resemblances ever since. In a sense, this was a pre-adaptation for study of Darwinius, which came much later. In the meantime, I got interested, again, pursuing the idea of change through the geological record in the fossil record. I got interested in the transition from the Paleocene to the Eocene epoch. This is the time when true primates first appear, when horses first appear, when the split-hooved ancestors of cows, sheep, goats first appear, and a number of other orders. Bats first appear, whales first appear, and really focused on that interval. Soon, within a few years, it became clear that where the animals changed, and they all changed basically at one bed that we could identify in the field, where that happened, there was a major isotopic event marked chemically that in the end was explained as a global greenhouse warming event called the Paleocene-Eocene thermal maximum. In studying fossils up through time in the geological record, I became interested in quantifying evolutionary rates to answer the question, how fast is evolution? The answer, contrary to Darwin, it's the one thing he got wrong, the answer is evolution can be very fast. So coming back to the global greenhouse warming event, I think that primates evolved, true primates evolved in that event. I don't think we will find the stem lineage earlier in the Paleocene. I think they evolved when the event started. Now, I've since spent 20 years, I'm sure I'm best known for studying fossil whales, and one of my favorites from Egypt is mounted right here in the Senckenberg Museum, so I feel right at home.
Time to stop.
So I'll stop.
Thank you. You can continue later.
Yeah, I'm Jens Franzen, and I'm now honorary member of staff of the Senckenberg Institute. The situation was quite different 36 years ago. At that time, it was really stressing. After more than a year of fighting, we got finally the permission to undertake excavations at Grube Messel, and due to the efforts of the Senckenberg Society, we also got the money and even more money from the Volkswagen Foundation at that time. So the permission came at the beginning of June, and already 4 days later, I began with an excavation with about 60 colleagues from our house. Many were very, very interested to come into Grube Messel and to help to rescue as many fossils as possible, because the locality was heavily menaced by plans at that time to install a large garbage dump for the whole Rhine mine area at that place. So we began our diggings, and we were really happy to find, among quite a lot of other interesting things, almost complete skeleton of a horse, of a dawn horse, a tiny horse, size of about a fox terrier. And this became really important for paleontology, because it turned out it was the first fossil horse that contained gut content. And then I realized how wonderful it would be to get one day such a skeleton of a primate, of an early primate. But we had to have patience, we had to wait until 1979, when Wighart from Koenigswald has come to Darmstadt, to the Hessisches Landesmuseum, and discovered during the preparation of fossils from Messel that one of those was in reality a primate, but you will come back to that, Wighart, I think, later on, and it was fragmentary. And Wighart also said already at that time, yeah, there is a grooming claw, and therefore I think it should be related to lemurs. So then time passed by, and it was 1982 that we got the first primate from Messel, which could be determined down to species, because it comprised the skull and also the dentition. But the dream left that to find one day a complete skeleton of a primate. All what came out were fragments, 7 fragments. And I had to wait for my 70th birthday, and then Ida, or scientifically Davinius Marcille, came into my life by an email from Jørn Hurum from Norway, which was accompanied by a photograph, and this showed a primate, just a dream of a primate for me. And this is a part of our conference today and during this week.
Okay, thank you very much. So, you now know the players on the stage. Now we need to explain to you something of the players in the fossil record. The first question that I'm going to raise, I think I will give a short background so that if some members of the audience were not in the session this afternoon, it will introduce to them the topic that we're thinking about. This topic is the origin and diversification of the modern primates. There is an argument, as has been mentioned already tonight, whether primates from the Paleocene, earlier than the Eocene, are in fact primates or near, or perhaps not so near, relatives of primates. But we know from the living primates that there are several major groups. There are the tooth-combed lemurs living on Madagascar and lorises of Asia and Africa. There is the tarsier, an isolated relict form living in Southeast Asia. There are the anthropoids, including the monkeys of South America, the monkeys of Africa and Eurasia, and the hominoids or apes, of which we are one. There are questions which have been raised in the past about how to best divide and discuss these living primates. For many years, there was a comparison between prosimians, including the lemurs, lorises, and tarsiers, versus the anthropoids, or a dichotomy between strepsirrhines, the tooth-combed forms, compared to haplorhines, anthropoids plus tarsiers. Genetic evidence, soft tissue, hard tissue, and many other lines of evidence have been used to attack this problem. And we now, I think, would all agree that on the one hand, we have the strepsirrhines or tooth-combed forms, on the other hand, the anthropoids. So the real question is, how do the fossils, especially the fossils of the Eocene epoch, during which Messel was laid down as a fossil site, relate to these forms? And I'm going to ask to start this question Dr. Xijun to give us a summary of some of the work he has done in terms of understanding this differentiation, and then there will be some other people who will comment on his statements. So,
first, I have to say the so-called strepsirrhine and haplorhine is a feature related to nose. The stripe means something twist, and the rhine is nose. And the haplorhine is the simple single, it not means a single nose, but it's a simple nose. Very unfortunately, we cannot find a fossil nose. Even in Messel, like a very beautiful fossil like Darwinius, no nose are preserved because nose are soft tissue. We are hoping in the future we can find some impression of the nose. But by now, we are talking about strepsirrhine and haplorhine or some other features. So it's not directly the feature of nose. It's in terms of the phylogenetically. That means which taxa is more close to the, we know the strepsirrhine, and which taxa is more close to, we know the haplorhine. So in my study, I would say the adipiforms that are kind of extinct lemur-like creatures that are more close to the lemur, tooth-combed strepsirrhines, and omomates, anthropoid, and tarsiers, they belong to another branch. It's called haplorhine.
Mark, would you like to expand on this a little bit in terms of what we can say about Eocene primates that might be related to the different living groups, and then we'll ask other people to comment as well.
Yes, however, what I would point to is that haplorhine are defined by a series of important anatomical characters which have been defined, and so they look derived and can be really defined by a lot of derived character, anatomical character, post-orbital. The post-orbital septum would be very important if it were the same in tarsus and in simians, in monkeys. However, there are good arguments that might be a convergence. And in all the fossils we have not the beginning of a real post-orbital septum, neither in omomates nor in all the adapiforms, which show that sometimes the evidence which would make us very happy and point clearly to some relationships is often not available. Now, the adapiforms have been found lemur-like in many aspects of their anatomy and also their postcranial anatomy, and probably they are quite derived in enhancing the opposition of the thumb and the other digit in the hand and in the foot in having very prehensile foot. And probably the character of the tarsal bone that we discussed this afternoon are related to an increased reliance on opposability of the big foot, and that can be traced back very far away. However, when we come close to the early Eocene, the differences are much less clear to read in the fossils, and so the fossils make it difficult. And as I explained before, the group that I know well in Europe are not very closely related to the living forms because they make big radiations and became extinct.
Who else would like to comment? Phil, do you want, or Chris? Okay.
Well, I think it's important for people to understand that there are certain groups of animals for which we have an excellent fossil record. We can trace lineages backward through time. We understand very well their history back even 50, 55 million years. Horses come to mind as a group that has an excellent fossil record, part of it at Messel, but in many other parts of the world at the same time. Whales come to mind as a group that has an excellent fossil record that enable us to trace things backward and forward through time without very much controversy. Primates have an excellent fossil record, but it's not as well spaced as we would like. It's not as continuous as we would like. It's very good in certain intervals and not so good in others. It mostly consists of jaws and teeth that are informative to a specialist, but not very informative to answer general questions. So much of our understanding of primate evolution is based on interpretation, either morphological or biochemical, molecular DNA interpretation of living animals and projecting the history, but making up a history for them from the present in a sense, and then trying to understand how the fossils we have fit into that. And this is where the Darwinian fossil is unusually important because of its completeness. It's partly beautiful, and in being so complete is a kind of icon of what we wish all fossil primates were. And so it's important in that sense, but it's also important in connecting all the parts of the body together in one animal so that we know what kind of ankles go with what kind of skulls, go with what kind of forelimbs, with what kind of foot structure, and tying that all together without having to guess or make inferences about it. So it's important to understand parts of the fossil record are better and less controversial than primates are. Of course, primates are close to us, and so we're particularly interested in them. And so even though we don't always have answers, we will argue about the issues, trying to resolve them because we ourselves are so interested.
Realize that in the Eocene, I don't think it's been clearly stated, there are these two major groups of primates. The adapiforms on the one hand, which tend to be larger and have often been thought to be related to lemurs and perhaps other strepsirrhines, and the omomyoids or omomyiforms, whatever term one wants to use, for a group which is mostly smaller, some of whom have large eyes and often are thought to be related to tarsiers. And the question is, can we make those relationships clear, or in fact, are things much more complicated? So let me ask Chris to talk next, and then...
This question about the initial differentiation of primates, and especially the origins of the two major taxa, Strepsirrhini and Haplorhini, is incredibly interesting. It's a very active field. But I guess I come down on the side of viewing the Eocene radiation of undoubted primates as already showing that that differentiation had occurred. I think that it's relatively easy to ascribe most Eocene primates to one or the other of those two suborders. Let me give you what I think is maybe the best example. Tarsiers, with the exception perhaps of us, are probably the most autapomorphous primates on the planet today. Alfie Rosenberger's presentation showing you how big tarsier eyeballs are is one example of that. Animals with those many clearly derived characters, I mean, these things are guideposts, these are icons that we can latch on to and say, okay, if I can make something related to a tarsier, then that gives me some evidence about morphology, about tree topology, and about timing of cladogenetic events. I'll nominate Shoshonius cooperi from the early Eocene of Wyoming, an animal that is known by a series of nice skulls, major limb bones, ankle bones. For an Eocene primate, it's one of the better known animals we know about. If that animal is not a close cladistic relative of tarsiers, I give up. And if it's a close cladistic relative of tarsiers, then it has many, many repercussions up and down the evolutionary tree that we can discuss.
When we began the investigations on Ida, we were more or less convinced at that time to work on a fossil lemur, some kind of a fossil lemur. Although on the other side, there were already some indications in one of the skulls we got of so-called Europolemur regarding the front dentition, for instance, to think of a possible anthropoid relationship. And in 1994, we had a wonderful conference on anthropoid origins, and there this theme was extremely discussed, and I wrote at that time what we would need are more complete skeletons in order perhaps to get to some kind of a solution of that question. And now we have Ida, and we can see that traits which should be there, which should be characteristic even of a fossil lemur, like a tooth comb or a central incisal diastema, are in reality not present. And the dentition looks really in the front very much resembles very much that of anthropoids. So the lower incisors are not procumbent, but rather vertical. And the central lower incisors are rather large, larger than, smaller than the lateral ones, whereas in the upper jaw it's different. It's just the other way around. So these are questions we can tackle, we can go on with the documentation as we have it at hand now at Messel.
I think there are two things that I'd like to say, one quickly and the other might stir up some controversy here, but why not? I'd like to think that we can sort of shift things around. I think we're at an interesting point here where things can change for the better and we might be able to learn some new things. We've been stuck in sort of a silo for about 20 or 30 years now, identifying these groups that Erik laid out, these Eocene groups, the two different groups, the Omamide, let's just call it that, the Adapiform, let's just call it that, as distinct groups, thinking really that they're natural groups, what we call monophyletic groups. And we've been trying to kind of look at them horizontally, can't look at them vertically through time, but comparing these Omamides to anthropoids, etc. We now pretty much know that those groups aren't really groups. The implication, natural groups, they can be subdivided, probably. Certainly the Omamides, perhaps even the Adapiforms as well. Once we do that, we might have found that stem group, perhaps, that would give us some new information that we can be looking at in order to trace the origins of these higher groups. And remember that the higher groups are usually major adaptive breakthroughs. Big things happened. We don't know if they happen quickly or they happen very slowly, but they're major jumps in evolution, in morphological features, in adaptation, et cetera, et cetera. And the older models just weren't really giving us the right perspective to get at that. The other thing I'd like to say is what's happened with Ida, in a way, is turning the clock back. Because during the 1970s, we really began to develop these strong ideas that the Adapiforms from the Eocene were related to the modern lemurs and lorises, that there was really a distinction between strepsirrhines and haplorhines, and it was a very fundamental distinction in primate evolution, with two very differently adaptive major categories. Ida, or Darwinius rather, I see it's over there, I'm not done.
The button comes in 10 seconds.
What does Obama like to say? We are renegotiating or revisiting the very same questions that were basically settled 30 years ago. And I'm not sure that's a very good place to be.
Okay.
Yeah, well, first of all, I just wanted to emphasize that for the audience, in case it's not clear, the fact that we can all of us can accept that there's a haplorhine-strepsirrhine dichotomy is actually a breakthrough. And we're actually finally all operating within the same phylogenetic framework. That's great, because what we do with that framework can be totally different, as we saw today. But I just wanted to point out quickly, we're looking to some of the big questions in early primate evolution. In my opinion, one of the, aside from not having any stem primates, potentially, one of the other really huge questions is where omomyiforms or omomyoids fit in. And this has profound implications for our understanding of early primate evolution. So there are two models. I mean, we've heard one, but there's another model in which tarsiers and anthropoids are more closely related to each other than to any of the extinct omomyiforms, and that's a position that's been put forth by people like Rich Kay and Callum Ross, Matt Cartmill, and if that's the case, then there is a long sequence of not only soft tissue but hard tissue characters that would support the monophyly of haplorhini. If tarsiers are related to omomyiforms, if they're nested within omomyiforms, just about all those characters break down as being synapomorphies of haplorhini. One of the things that we've seen with over the last, say, 20 years is it's been very difficult using genomic evidence to resolve the position of tarsiers. There's a paper published just this last year that I think kind of solidified that tarsiers and anthropoids do form a clade. But still, it was a very, very basal split. And in my phylogenetic analyses, I'm actually fairly agnostic about this. But in my phylogenetic analyses, and also in what we saw today, omomyiforms and tarsiers are coming together, and that really complicates the picture of haplorhini, because then at the base of that clade, there's literally very little morphological evidence. And so that's a major question in early primate evolution.
Okay, so we've raised a number of problems about this early diversification. Let's take a look now in a little more detail at Ida, at the Darwinius fossil and the problems it has raised, having the background of concepts and taxa to do this. And I'll ask Jens to comment a little bit on how this fossil was found, how he first became involved with its study, but also I'll push you a little bit to say something about the fact that you also described the counterplaque as a completely different animal with some other interpretation.
Right. This is a good example to show how important the completeness of the documentation is. Because this opposite plate didn't contain the hands and the feet, and the length of the anterior limbs and the length of the posterior limbs. And so the situation changed completely having plate A at hand. Then it turned out that the proportions of the legs were absolutely different from that genus Godinotria, of which I thought before that Darwinius could belong to. And so this was the beginning, the rethinking on the systematic position of Darwinius. And also, the deeper we were investigating Ida, the more we found characters or evidences, structures, which seem to have more affinities to haplorhine primates than to strepsirrhine primates. So we changed our mind during those investigations.
Okay, thank you. I'll ask Professor Gingerich to comment a little further. Let me raise one point which Jens touched on earlier, and Phil, you may want to comment on this or ignore it as you decide. But when we look at these Eocene fossils, as several people have said, they are clearly not definitive members as a unit of the crown group. So, for example, if we look at Eocene adapiforms, we think they are a sister group to the living strepsirrhine lemurs and lorises, not necessarily living tooth-combed forms. We don't expect them, at least I wouldn't, expect them to have a tooth comb or to have other derived characters which characterize the living forms because they are earlier in time, they have not yet developed those features, and no animal which has those features is known until the ones that Erik Seiffert mentioned earlier that he described from early levels in the Fayum 10 or 15 million years after the beginning of the Eocene, or even later than that. So therefore, one has to be careful what you expect to find, and if you say, well, we didn't find that, it proves A, well, perhaps you shouldn't have expected it because that wasn't the right question. One has to think about that. And on that basis, let me give you 3 minutes, Phil.
Well, I think it's important to remember that we live in the present. We're familiar with the animals that live today. Those are the ones we see. Those are the ones we go into the forest and study. And as biologists, it's very easy to imagine that that's all there is, that that's all there ever was. I sometimes chide my biological colleagues as thinking that life in the past was just like today, there just wasn't as much of it. And it's important, I would say, if I've learned anything as a paleontologist, it's not to expect what we see today in the past. Evolution is real, life has changed, dinosaurs have come and gone, and the same is true in the Eocene. There are groups of animals there that we don't have with us today. And that includes the primates, the tarsier-like primates, the omomyids that we've mentioned here, are a group that came and went. Adipoids are a group that came and went. But somewhere in the Eocene, something survived and became the Oligocene primates, that became the Miocene primates, that became the Pleistocene primates, that became even us today. Something in the Eocene is real, and something in the Eocene is ancestral. And the question is, what is in the Eocene? And given what's there, what is most like the higher primates, if that's what you're interested in, or what is most like the strepsirrhine lemurs and lorises, if that's what you're interested in. And so much of what we've done with Ida, with Darwinius, much of what we do with other fossils is study them for what they are, and then put them in some kind of bigger context, some kind of numerical categorization of their morphology, and try to understand what's primitive, what happened early, what's derived, what came later, and group animals based on their derived similarities. So that's what we've done with Darwinius, and as I talked about today, when you do that, you wind up with features of Darwinius. I started with a catalog of 30 features, and only 6 in the end proved informative. So something like 5 out of 30, 6 out of 30, 20% were informative. But the ones that were informative were consistently like the higher primates, like anthropoids, and there were none that link Darwinius to strepsirrhines. I think if we took any of our data matrices of different sizes, different complexities, took all the fossils out, I think we'd get the same topology that would separate Haplorhini and Strepsirrhini, and we would all agree.
I'm not quite sure that we really know what was living at Eocene times. So if you say one of the known Eocene fossils have to be ancestral to the anthropoids or the haplorhines, then I would say be careful. There may be more material in Africa we do not know so far. On the other hand, we see characters and they are evaluated very differently. I think I could show that there is one character in Darwinius which is very close to that what we see in modern lemurs, the grooming claw, which is very different from other grooming claws. And I would, from that study, refuse the idea that just grooming claws are primitive. I think human claws developed several times independently. And so I think we have to follow up the history, and if it is reasonable or not that specific characters changed during time.
Well, let me just answer that very briefly, if I may. I agree very much that what we think we know is always in the context of the record we have, and there is always lurking in the back of our mind the worry that maybe the things we're really interested evolved in the middle of Antarctica, which wasn't yet covered with an ice sheet. Right. So there's always a possibility that there are things we don't know that would be important.
I'm not going to say anything about Darwinius. I would like to go back to what we started talking about a few minutes ago, though, because we all seem to have rushed past the notion that we have no idea where primates came from. We all started to jump into where anthropoids came from. I still want to know where primates came from, folks. I mean, we didn't really talk about that. Chris pointed out that early in our record that we know, as far as we know, strepsirrhines and haplorhines are there right at the very beginning, as far as we can tell. But I think that masks some other bit of information, and that is that if you look at the very beginnings of both of those groups, they're very subtly different. They're not tarsiers and lemurs sitting there in the fossil record at the beginning of the Eocene. And while we may not know what the basal sister taxon is anymore, if it's not Plesidaepiforms, then it must be something else. I would like to sort of have a sense of where primates could have come from, if not from Plesidaepiforms. Maybe they have. Maybe Phil is correct, and they just evolved very rapidly from something that we haven't really been able to identify yet. That's perfectly possible. Maybe they came from Antarctica. I don't know, but I think we rushed past the origin of primates to get to the origin of anthropoids, and that's a tad of a concern.
Yeah, I'm not going to win any congeniality awards for saying this, but I think some people have been dancing around the subject. I'm just going to stand up and dance. In my opinion, Darwinius is probably a lot like a Picasso painting. It's beautiful. It's probably worth a lot of money, but it may not mean anything. And what do I mean by that? I mean, does it really tell us anything about primate evolution that we didn't already know from Notharctus? I would suggest so far it doesn't. It reconfirms what Notharctus already told us. The only thing it has that Notharctus doesn't have is some stomach contents, which are nice. But I think Alfie kind of hinted at this a while ago that, we've kind of reset the clock, we're back to the future. And Darwinius, it's a beautiful, beautiful fossil, and congratulations to everyone who found it, studied it, especially the people who currently have it. But I really don't think that it has been a game changer, and I guess that's all I need to say.
So let's go in quickly. Wighart will make one statement quickly.
Just
then
one statement. If you say there's nothing new, but the confirmation of things which are isolated in Notharctines, here in one individual, that is very important and something new.
I would follow that argumentation. Ida, of course, it's beautiful, but it's full of informations, not only in a taxonomic sense, but also in a paleobiologic sense and in a functional sense. It opens up the possibility to really understand what we call characters. What are characters? Characters are not made for systematic purposes. Characters, sorry? Yeah, characters are not made for systematic purposes. Characters develop as necessary structures for the survival of their bearers. And so we have to think of the functions, as Alfred Rosenberger did already earlier on this morning, and in order to find out what structures belong together to one functional system. And when we are coming to that point, we have much stronger arguments than just with a simple, small structure.
Yes, we mentioned a few of those characters, and we would like to understand characters. But you mentioned, for example, the vertical incisors that we found in Ida. This is known since a long time in many adapiforms, but this is believed by almost every primatologist that this is primitive in primates. So how can you make an argument that this is shared-derived and indicating anthropoid relationships? No, it has always been considered as primitive in primates. And I mentioned another character that Philip has on his list today, fused mandibles. Fused mandibles are found in different groups of primates. They occur in later, within adapines, they occur in adapes, become fused very young, but no one would ever suggest that adapes is monkey-like. I mean, there are a lot of convergences in the characters. And so the important aspect with the characters of Ida would be, can we make sure that some characters are really shared-derived and meaningful in terms of haplorhine or similar to anthropoids. And the characters you have listed are just, similarities. Many of them are primitive. They might be interested in terms of function, but in terms of real phylogenetic signal, they are lacking. It's a good adapiform. It fits in that radiation.
I just didn't want this to get lost because I mentioned it in the introduction. And I think that people tend to focus on the haplorhine side of the tree, and the strepsirrhines are getting lost. And as I mentioned, we're still dealing with primarily jaws and teeth, but we're making progress in our understanding of the early evolution of strepsirrhines. And I want to remind everybody that in 1997, Elwyn Simons described two fossil primates from Egypt as circumaniene adapiforms, based on some partial mandibles. Those purported circumaniene adapiforms, when we found the front of the mandible, almost certainly had tooth combs. One of them is Wadi lemur, and it has these very procumbent premolars that are what you find in a galago. So, I mean, based on the teeth, what we thought was an adapiform, and it was a perfectly reasonable interpretation, is actually a full-blown crowned strepsirrhine, in my opinion. Now, also, from the fossils that are being found in the early Eocene of North Africa, and from the fossils that we found from BQ2, when we put those into a phylogenetic context, forms from the early Eocene like Jebel lemur and what was described as Oncomomes from Africa, again, what's otherwise a circumaniene adapiform from Europe, these come out as consecutive sister taxa of crowned strepsirrhini. So actually what we're finding is that the earliest, the things that are right near the base of crowned strepsirrhini could have teeth that could very easily be mistaken for circumaniene adapiforms. And just to remind you, Darwinius is a circumaniene adapiform.
And just to remind you, it doesn't have a tooth comb, it doesn't have procumbent incisors, it doesn't have pointed incisors.
Yet.
Coming back to Mark's point, the characteristics of the incisors are part of the cladistic analysis that I presented today. And in the cladistic analysis, they come out as being, the spatulate incisors come out being derived, primitive incisors come out being primitive. I didn't make it come out that way. That's the way it comes out. And so I don't think you can turn around and say and think that spatulate incisors are primitive for primates when treasurers have pointed incisors, Plesidapiforms, if they were to be related, have pointed incisors, tarsiers have pointed incisors, lemurs have pointed incisors, lorises have pointed incisors. Nothing has spatulate incisors until you get to adipoids and anthropoids.
Dehardina asiatica have speculated lower incisor, doesn't have a pointed incisor.
This is good, and we may be able to come back to some of this, but let's move on to another question that has been raised. This is about the origin of the sort of next interesting group of primates, the anthropoids. As we said, anthropoids. And I was asking the other day, there is unfortunately no German term for what we call in English monkeys. Affen is a much broader concept. One can have specific kinds. Pavianen. Ja, aber für Katharinen Primaten von Papio, Macaca, Schlankaffen, es gibt kein Wort für die ganze Gruppe und auch für Platyrrhinen von Südamerika. I'll stop with my German at the moment, but in any case. So the question now is, we don't know where modern primates came from. We don't completely agree how those earliest primates diversified. So can we say anything about where so-called higher primates, anthropoids, whatever we want to say about tarsiers and omomyids, we put them aside for the moment and concentrate on the origins of the group which today includes the South American monkeys, the African and Eurasian monkeys, and the hominoids, apes and humans. So what can we say about the origin of that group? And I'll ask Dr. Rosenberger to start, and then Dr. Seiffert, and then we'll all come back and fight with each other again.
I think, first of all, it's important to think about what the adaptations in general are of I'm sorry, thank you. At least the cranial adaptations in a general sense. It seems like anthropoids fundamentally have reduced the sense of smell and probably, perhaps, have changed something in the visual system, and there might be a trade-off there in terms of what sensory system is going to be the predominant sensory system. Anthropoids also are diurnal, and we don't know, fundamentally diurnal. We don't know when that adaptation first arose, if it was in the origins of anthropoids as some sort of a major transition of living during the daytime, making a living that way, or if it was in an earlier trait that they just inherited from some other group of animals. And we don't have very good functional interpretations of some of the other structures. We know something is going on with the orbits, for example, as I was talking about earlier today. Their orbits are sealed off entirely from the rest of the skull, and we really don't have very good functional interpretations of that. It may have something to do with mastication. It might have something to do with the development of the head and the bones in the head. It doesn't appear that anthropoids at the very beginning were any smarter than their other non-anthropoidean primate relatives. So I'm not sure, I don't think that there was a change in cognition, although maybe in processing information, because they are, if they're processing visually, all mammals that have a strong visual orientation and process a lot of visual information tend to have relatively large brains. So there's sort of a paradox there that I don't really comprehend. So those are sort of some of the adaptive gestalt that we're dealing with. The locomotor system seems to be fairly generalized, we think, as we reconstruct these behaviors and anatomies by looking at bones. It's very difficult to say what was involved here. It might not have been such a big thing. It could have been just sort of, they were successful. Darwin told us how that works. Platyrrhines versus the others would be an interesting question, but I don't know that we'll have time to go there. Yes, well, I may as well give a personal advertisement here, because I did emphasize Runia today. I think, to me, I'm very cautious about this in a sense, because I don't like the idea that we only have one specimen. And this seems to be a very weighty hypothesis to be pinned to one single specimen, this Texas late Eocene Runia skull. But it happens to be an extraordinarily beautiful, complete specimen, and it's rich with information. And as one of the things I meant to say this morning, and I don't know that I did, was enigmatic because no one has really studied it well. It's been sitting there for a really long time. And so hopefully we'll get more out of it. That's very valuable.
Okay, so on the origin of anthropoids, I'll talk a little bit about just the fossil record, and I think it's important to start with what I think we can all agree on, which would be that some of these fossils from the right around the middle late Eocene boundary in Africa, like Baratia, Proteopithecids, some undescribed material. These, so we can't agree on that yet, but these are the earliest undoubted anthropoids. And if you don't think that those are anthropoids, let me know. But okay, we all agree on that, because before that, I mean, going back in time, I think it gets very messy. What we know from these early African anthropoids is that they were very small. They were probably frugivorous, possibly to some extent faunivorous or insectivorous. And what's remarkable about them is that many of our conceptions about what makes a higher primate, what makes an anthropoid, actually breaks down when we start to look at the fossils. So in Flegel's textbook, we learn that the features that define anthropoids are things like fused mandibles, big brains, the absence of paraconid cusps on the lower molars. These are the anthropoid features. Well, when you look at the earliest undoubted anthropoids, and in my opinion, the oldest crown anthropoids, that I think are crown anthropoids, all those features are absent. They have small brains, they have unfused mandibular symphyses. There's not an Eocene anthropoid that I am aware of that has a fused mandibular symphysis. And a lot of them have paraconids on all the lower molars. Okay, so there's been a tremendous amount of convergence right at the base of Anthropoidea. Do I have time to keep going?
No.
All right, I'll come back to it.
Yeah, sorry, but that's okay. Chris, do you want to comment, and then we'll see who else wants to.
Yeah, I basically endorse everything Erik said. I think that, in fact, these highly primitive yet undoubted anthropoids from North Africa, again, are excellent signals to us that the Eocene anthropoids were not modern anthropoids. This is the same question that that early 20th century paleoanthropologists were trying to settle with respect to human origins. Did bipedalism evolve before big brains or vice versa? We're at that stage with anthropoid origins today. However, I think it's incredibly important that we have to frame everything properly. What is an anthropoid? What do we mean when we say anthropoid origins? If we agree that tarsiers are the living sister taxon of anthropoids, then I'm going to call anything an anthropoid that is more recently related to modern anthropoids than it is to a tarsier. Okay? If your phylogeny is correct, which I highly doubt, but without that kind of a definition first, people start talking past each other by saying, it doesn't look like a monkey, so it's probably not an anthropoid. We don't know. I mean, Sahelanthropus probably doesn't look like a homo either, but we can debate what it is phylogenetically. In that sense, I think that Shoshonius is an incredibly important guidepost in reconstructing anthropoid origins. That animal is 50 million years old. It has many, many synapomorphies with living tarsiers. If it is we can even call it a tarsiid if you like. If that animal is a tarsiid 50 million years ago, then Erik's 37 million year old undoubted anthropoids from North Africa have been evolving for at least 13 million years. And there's a big gap in the fossil record where we do not know much about what anthropoid anatomy would have looked like.
Okay, Phil? Watch the clock.
Who are you talking to?
You. I just said Phil, so you're talking.
Well, I'm going to play the devil's advocate.
Or just the devil.
Erik.
Go.
I didn't hear that, Erik. Erik Seiffert has focused on Baratia as the model for anthropoid origins. I'll remind you that from the same beds he described a larger primate that has many of the characteristics of anthropoids that he had to explain away as convergences. We don't know the skull of that animal. We don't know the skeleton of that animal. Again, playing the devil's advocate, I'm going to suggest that maybe when we find them, they could turn out to be anthropoids. And so I think we have to be careful to not be too selective in terms of what we focus on as being anthropoid and what we dismiss as being convergent with anthropoids, and accept that there's some uncertainty here, and we might all be surprised. Now, I'd like to ask a couple other questions. Erik, again, Seiffert, if you've described tooth combs in Wadi Lemur, it would help if you, let me ask, first of all, are the tooth combs associated with the jaws? No. It's an inference, okay. And it would help if these were illustrated in a way that was convincing. I just learned here that Tyardina has spatulate incisors. Are these described in a way that I should know about them? This is published or not published?
We mentioned this, but not fully published, not fully described.
I wondered. Well, I look forward to seeing the publication of spatulate incisors in Tyardina, because I would be surprised.
Yeah, we actually score it in our data matrix, and we put this in the supplementary data of Nature. Unfortunately, people usually don't look at our supplementary data.
Well, I would think that's important enough to bring up front somewhere and illustrate very well to make sure we don't miss it.
Okay. So
yes.
Do we have someone else at the table who has not? Gregg, do you want to?
Yeah, I just want to add that Erik Seiffert pointed out many of the animals and many of the features that we look for in anthropoids don't really exist in these very earliest forms that we're recognizing as basal anthropoids. And I think Chris and I have talked about this in the past, that you may not actually expect those animals to have most of those features because they are very primitive. And I think it still doesn't get us to the origin point, because I think we're looking maybe in the wrong place if we're looking in Africa. I think it's much more likely that we're going to find, and maybe I'm stealing your thunder, but the fact is that we probably ought to be looking in Asia for something that ultimately gets to Africa to produce these things that become, crown anthropoids eventually. I don't know. I'm not ready quite to endorse Eosimius as the thing, because I'm not sure we'll I don't think we'll actually ever know for sure, phylogenetically speaking, what the animals are that were the sister group to anthropoids, because I don't think we can recognize them in any kind of morphological way. So it's just going to be more of maybe a STEM definition like Chris has proposed for Shoshonius and the things between it, or maybe we should just drop the phylogenetic definition completely and look for some ecological way to recognize these animals as starting to behave like things that you might expect to lead to an anthropoid. That's all.
Okay. Before we start, I'm just going to say we'll give Phil and then Erik 1 minute each for a comment. This is how the presidential debate should be run. You've got to be tough out here. So when says 59, you stop.
I forgot my third point.
Well, Gregg reminds me of another question I wanted to ask Erik Seiffert. Baratia is a model for primitive anthropoids. Where would you put the Amphipithecus and its relatives? They're more or less
That's not a 1 minute response.
Beg pardon?
It's not a 1 minute response, sadly. But.
That's not a 1 minute response?
Well,
it's a yes or no question, so...
Under the table. Next.
Okay.
Because they certainly don't fit the model of Baratia as the only morphology of a primitive anthropoid. No, if some people like Chris Beard have argued that they are anthropoids, some like Gregg and even I at some points have argued that they are adipoids. They're a good example of something in the late Eocene that's very difficult to place on one side or the other.
Yeah.
And none of them look like Baradia.
No, they're very, very enigmatic, highly specialized, and if I had to put my money on it, probably very, very specialized stem anthropoids that have become huge and have, you know, teeth and jaws that reflect that.
And sure look like adipoids.
Okay, now wait.
But you brought up the point of Aphoratopus, and I know that, you know, an important part of the hypothesis that anthropoids arose from adipoforms, it comes to Africa. This has been talked about. And so you would expect something like Aphoratopus to look very anthropoid-like, and in its dentition and its mandibular morphology, yes, it does. It looks very anthropoid-like. We do have postcranial remains. Sadly, the only one that has been published so far is an astragalus of Aphoratopus that among primates looks more similar to a loris than to anything else. It is absolutely not anthropoid-like. And I'm fine discussing unpublished fossils. We do have others, and they just don't look very anthropoid-like in the postcranium. They're not becoming more anthropoid-like.
And these are associated with the jaws and teeth in the way that Darwinia's jaws and teeth are associated with its ankle bones?
No, but fortunately Aphoratopus is by far the largest primate in that fauna, so we can be pretty confident about the attributions.
Okay, thank you. And Chris, all right, let Chris make one statement and then Mark. I didn't.
Yeah, just in defense of Amphipithecids, a couple of points. One is that, okay, Baratia, bear in mind, or at least I, and I suppose Erik, are endorsing it as a late Eocene anthropoid. That doesn't mean it's a morphotype for what all early anthropoids look like. It just means it's one of probably a large diversity by then of late Eocene anthropoids. More diversity can be found in Myanmar. If you look in the Pandong Formation, you've got a big diversity of amphipithecids. You have other animals called eosimiids that live there. Almost all of them are certainly anthropoids, but amphipithecids we can put to rest tonight. I don't know what the ambiguity is. There are ankle bones from Myanmar. My colleague Laurent Marivaux and several of us have worked on these and described them. They are anthropoid ankle bones. They are the right size to be amphipithecids. So far, there is no other dental taxon in that formation that can pertain to these ankle bones. So I don't know, I don't know how we can twist into a pretzel to claim that these animals are not anthropoids. They have ankle bones that look like anthropoids. They have teeth that look like anthropoids. If it walks like a duck and quacks like a duck, it's a duck.
Thank you. Mark.
No, I do not find that their teeth look really anthropoid-like. The talus look also like that of homomites. I mean, it's no? Like a monkey. And is it primitive or is it derived in the monkeys? Well, a big discussion that we have and an argument I have with Erik Seiffert is this model of anthropoid origins as having reverted many characters of so-called prosimians, and I find this extremely unlikely at the beginning in a radiation that one group would revert a lot of these postcranial characters in primates. I find this very hard to believe. In fact, I don't believe it at all. Concerning the teeth, when I looked at the teeth carefully of, Prondongia and Amphipithecus for the 94 book, I was struck that they were very, very bunodont and proposing that they were catarrhine. But then a few years later, were found the much more complete dentition of Siamopithecus. Now, if you look at the upper molars of Siamopithecus and of all other anthropoid of amphipithecids, you never see a complete crista obliqua on the upper molars, and the crista obliqua is shared by platyrrhines and by catarrhines, and I would put that really as a typical dental character of crown anthropoids. So they don't look any more anthropoid-like to me. And the last reason for me to be very, very reluctant with eosimids and amphipithecids as anthropoids You know, anthropoids are a very, very successful group. They're very, very good adaptively. Asia is the refuge of extremely primitive primate and even sister group of primates like tree shrews, like dermopterans. They would have originated in Asia, and you would have no living anthropoid in Asia now, just coming out from their origin. It doesn't make sense to me that, you know, they would be anthropoid originated in Asia and not successful. Why is there no group of living monkeys of Asiatic origin? It doesn't make sense to me. And I emphasize that I like the historical approach. I want the history to be coherent, and on that side, it is not.
Mon cher, il faut cesser. Okay. It's now about 8:30. This has been very successful, but moved along farther than we had expected. We were going to discuss one more topic. I will ask the panel, would you like to go on to talk briefly about biogeography, or to continue this rather animated discussion about alternatives in anthropoid origins? I'm happy to go either way, but if we are to go to the question of biogeography, we could have 2 or 3 quick statements. It's now, as I say, 8:30. So we have 15 minutes. Well, we can only do that briefly, but if the question of the geographical position of potential early anthropoids has been raised by Mark. So we could talk about both briefly and say, all right, let's do this. Let's say that Chris can make a short couple of minutes statement about the importance of biogeography and some of the points that you made earlier, and relate them to anthropoids, if you'd like. And then we'll give each person a chance to have one very short comment about the combined topic of geographical and morphological origins of anthropoids.
Mark doesn't want anthropoids to originate in Asia. So my question is, if they originate in Africa, they originate from what? Anthropoids and primates, one thing that the molecular biologists agree on is that they're not afrotheres. What is it in Africa that can give rise to an anthropoid?
Well, this fits in.
Bear in mind that everyone agrees, including you, Philip, that the sister group of anthropoids is tarsiers. Where do tarsiers live? This can be a guide for where to search. In the same way that, that Charles Darwin, a man that many of us admire, used chimpanzees as a guide on where to search for human origins. It's much more parsimonious in a biogeographical sense to suggest that the evolutionary dichotomy between tarsiers and anthropoids, which occurred at one time and in one place, ultimately, it didn't happen everywhere, it happened somewhere, and it's much more parsimonious to suggest that this ancient event happened in Asia and that tarsiers were then relegated to Asia and anthropoids spread everywhere, as opposed to suggesting that this happened in Africa and, oops, something happened to the tarsiers, we can't ever find them, and something else happened to the stem haplorhines before that we can't find either. So, you know, I believe in the fossil record as much as the next person, and I would suggest that Asia is the place to look. And that's without bringing up the stem anthropoids that almost certainly are known from Asia, eosimiids and amphipithecids. I don't have enough time to talk about them in detail, but what I will say in response to Mark's comment of why aren't there any living stem anthropoid lineages in Asia, if anthropoids originated in Asia, then the initial colonization of Africa by anthropoids was a pivotal macroevolutionary event. It almost certainly sparked a huge adaptive radiation that Erik is picking up in the Fayum. And that evolutionary radiation itself may have sparked enough anatomical innovation, enough macroevolution that it transformed the bow plan of what was basically a primitive haplorhine into something that we would all agree is a monkey. Now that happened in Africa. When those things came back to Asia later, after the collision between I was asked a question.
Yeah, sure.
When those things got back to Asia after the collision between Africa and Eurasia, they reinvaded and wiped out whatever was left behind before.
Where did you get your information?
Deduction.
Okay, so we now have 1 minute. Mark, would you like to comment?
Very quickly. First, Shoshonius disappeared as the closest sister group of Tarsius, so it doesn't matter. And the fossil record in Africa in early time is very, very poorly known. We had a good demonstration today. There are things which are not known in Africa. And in Africa, there is Atlassius, which is extremely old, which is late Paleocene, and shows that there were primates in Africa very, very early. And there might have been something like a vicariance, something related to a tarsier coming very early in Africa, and there becoming, because it must have been a very long process. I don't believe in evolutionary miracles, making an anthropoid just after a crossing and invading a new area doesn't make sense. And so there is ample time and the possibility for something related to a tarsier, but still extremely primitive, reaching Africa, possibly related to Atlassius or somewhat later, we don't know, and all the differentiation of all the anthropoid character taking place in the early history of Africa, which is very poorly known.
Okay. Gregg, do you want to talk about either amphipithecids or?
I don't want to touch that, although I won't put them to bed yet. But no, I just want to, Phil asked the question about what happened to the anthropoids in Asia. I think this gets back to the notion of how we define origins. I think nothing happened to them, basically. They lived and died there, and had whatever it was that left Asia and got to Africa not gone there, we wouldn't be sitting here talking today. I think this is the whole point, and this is what Chris mentioned also, that it was a pivotal event. You know, if Eosimiids or whatever group you want to pick hadn't set sail and somehow got to Africa and went on its way, we wouldn't have higher primates as we know them today. Maybe they would have developed in Asia, maybe they would have not. Maybe we would never have higher primates. So I think that's, to me, the way in which you define the group is not simply based on a bunch of characteristics that you can find or not find, but what happened to them paleobiogeographically. Can you explain the origin based on some sort of scenario by which they leave wherever they were developing and get into a new place where they can really flourish?
Okay. Phil, do you want to?
Who says Eosimiids started in Asia? The first one was found and named there, but what is Afrotarsius? Is it a tarsier or is it an Eosimiid? Erik Seiffert, quick answer.
I would say, based on the recently described upper
your
teeth,
time.
I'd say it's a tarsier.
Okay, well there we've got a tarsier very early in Africa, and so I think it's an open question whether tarsioids, eosimiids, anthropoids, adipoids started in Africa or started in Asia. Is this the final 16 seconds I get for the whole thing?
Pretty much.
Okay, I would like to say that the most important, Darwinius is a very important fossil from Messel. It has started a discussion that won't stop this evening. But the work is not finished. I would like to see some kind of a tarsioid omomiid fossil from Messel, a skeleton like Darwinius, because we don't know that side of the family tree very well, and it would be very helpful to have a Messel quality fossil on that side of the tree.
And if I were Elwin, I'd pull it out of my pocket now. Those of us who were there remember that one. Do you want to?
Yeah, I think of the potential which maybe exists in a better understanding of characters. So just looking, what are they good for? What is their functional role within the whole construction? And then we are getting at an idea how high they may be developed, just showing somewhat the tendency of evolutionary development. And finally, we need to cluster those huge matrices of characters into functional units, in my view. Thank you.
Thank you. Okay. Alfred, do you want to take a shot here?
Sure. I'd like to ask one very simple question, and I want a show of hands from the audience. We've been performing for 11 hours. We like it, but perform for me. Does anybody here actually believe an early anthropoid crossed the South Atlantic Ocean on a raft to get to South America and become a platyrite? Does anybody believe that? Who thinks it's impossible and crazy? We win.
You finished? Okay. That's good.
I think that is almost a question that we should vote, where we would put Darwinius. No, I think that is not so bad. But I think the discussion is so lively, for there are so many gaps in between, and we try to bridge them by logic arguments. And I think the gaps are just too big that we could find one proper solution.
I just wanted to make
wait, just go around. So Xijun, if you would like, yeah. You're last, so you win. Except I come after you.
So actually, I want to say something about the characters, as I mentioned earlier this day, and the so-called characters. They existed in their nature. They certainly have functions. Why our human beings try to identify these characters? Because we need to understand this world. If we don't examine all these characters, how can we describe the so beautiful Darwinian specimen? We have to, to say this cost $10,000. So why do you want to reconstruct that tooth? You want to see the characters. That's my point.
Okay, sorry, I need to get back to Afrotarsius. I just wanted to make the point that it's now I think it's pretty clear from the recent work that there was an episode of faunal exchange between Asia and Africa involving two different clades of rodents, potentially multiple clades of primates, and this was probably later Middle Eocene into the late Eocene. And whether Afrotarsius is a tarsier or an anthropoid, I do think it's probably part of that exchange and wasn't part of the ancestral African primitive primate fauna. There's nothing before that.
That's all?
Oh, I get to keep talking?
You've got another
13 seconds. Well, right, yeah. So here's to finding more fossils in Africa. That's what we need.
All right. We're now 2 minutes from when the scheduled end of this project is, so I think rather than starting a different topic, I'll say just a couple of things to sum up. Erik and others raised an interesting point about the first presence of primates or rodents in Africa. There's just been, I haven't even seen it yet, but there's just been a very intriguing publication about rodents in South America several million years earlier than previously thought, and I heard that they're African rodents, not South American rodents, so I don't know the details on this, but it sounds like there's a lot going on in terms of biogeography and whether we should expect primates to turn up even earlier in the New World. The most important question, however, we always have to remember is we first have to do the morphology. Whether we do it in 3D or in pulp or on a piece of paper, if we're not talking about sister group relationships between two groups of organisms, It doesn't matter how similar they are in time or space. If they're both found in the good old Willwood Formation of South Elk Creek, or they're found in the Fayoum, if we don't know that they are each other's closest relatives, we can't ask about more detailed relationships. We have to look for the characters, especially derived characters, that link features and groups before we can worry about their transcontinental migrations. This was something I tried to say in anthropology 30 some odd years ago. It's the same thing in terms of ancestry. Stratophonetics is a wonderful idea, but as far as I'm concerned, only works if we know that we're dealing with sister populations, not just successive groups. We have talked about a number of methods that may be used in the future. I think that, as I mentioned last night, it's interesting that the study of primate fossils, including humans, has often been at the cutting edge of new techniques. I'm not sure why. Perhaps anthropologists and primate paleontologists are always running to catch up with people studying dinosaurs and whales and horses. So we use something novel to get our less important but still interesting fossils to where we can understand them better. And I think that there have been a number of different approaches discussed today which are important looking forward. We've got a lot of questions and disagreements which will keep us all happily busy and off the street until the next conference. Thank you all very much, and thank you again to the organizers for putting all this together.